Thanks to the organizers for having us here for this. This couldn't be done without my whole team, and especially Bama Nafsari, who unfortunately had a family issue and couldn't come to present for the group. He started out as a postdoc in my lab this summer and really took to the DREAM challenge, and it was a great project to get him started. So we've sort of been through the data, but again, there was the phosphoproteomic data from 4 different cancer cell lines, 8 pathway stimuli, 4 inhibitors measured over a time course, and the goal we focused in sub-challenge 2 was predicting the phosphoprotein expression in response to new inhibitors. Since we're a bioinformatics group, we started doing sort of the standard unsupervised analyses that we do when we get a new data set to look at what's going on before we applied any sort of prediction model for the leaderboard, which delayed our participation in the leaderboard a little bit, but gave us some insights, I think, in leading into the data. So the first thing we did was just a simple correlation analysis, where here I've plotted the correlation of each sample with every other sample on a heat map, and the darker blue represents more correlated one sample to another. And on the colors represent the stimuli along the columns and the inhibitors along the rows. And the first thing that we noticed from this in red, it's sort of hard to make out. This represents a histogram of the different correlations. And you notice that the correlation coefficient starts around 0.5, and the bulk of samples are really highly correlated one to another. We also noticed that the clustering is not the most, it's not doing anything to distinguish very clearly, at least in this cell line, between the different stimuli or inhibitors. So you get some separation of the AKT inhibitor, either alone or in combination, and the control with the FGFR inhibitor, but you're not seeing a whole lot of separation between them. We look across all of the other cell lines and notice a similar sort of trend where, again, you see a very strong correlation between samples and not much separation of the inhibitors. So we were sort of stuck at that point, not knowing exactly where to go from there. So what we did is we said, okay, this is something that I've been working on in my research independently. Clustering is not the greatest for time course data. I think everyone in this room can appreciate that. If I took just this simple toy example of a simulated data set, where imagine I have 3 main sort of conditions represented in yellow, black, and blue, and then I have something over time where there's something increasing over time in each of these sets where the shading represents different times. What's clustering going to do? Well, it's just going to find the things with the strongest signal that separate out the data, and it's going to mix together conditions and mix together time points, exactly like what we saw in the clustering diagrams before. If instead of clustering you use a matrix factorization approach, what does that let you do? In theory, then you could represent some basis here, represented in these patterns, where the idea is this basis represents across the samples what's going on relative in one another in strength in the different conditions and at the different time points. And then you can go and associate genes or proteins, whatever species, with those patterns. So for example, I think the easiest one to see if you guys can make it out is in this third condition. You see something that rises and falls right over this time point, and you end up with one basis vector that's able to account for that one rise and fall over that time point, which is then associated with this middle set of genes through a matrix multiplication. There's a ton of algorithms out there to solve that problem. PCA is perhaps the most famous. But the problem with PCA is, again, like clustering, what does it do? It finds the thing that explains the most variance in the data. So if I go back to looking at my data, what's the most variance? Well, the most variance is what's going up in all 3. But that doesn't necessarily separate one condition, one time trajectory from another. So instead, we've developed an alternative matrix factorization called COGAPS. And what that does is it ensures non-orthogonality of the matrix elements instead of orthogonality like PCA by using non-negative matrix factorization. It enforces that all the matrix elements have to be non-negative. And we also have a sparsity constraint that's imposed. It's not dissimilar to a lasso constraint, but we find our solutions using an atomic prior. And what that prior lets us do is is a cross between a penalty on the number of matrix elements and their magnitude. So it's an adaptive penalty so that if a matrix element is large, we end up with no penalty on that matrix element for being large. But if it's small, then you have a larger penalty. And it's enforced implicitly through this prior, which I'd rather not get into today. But we have implemented it in a bioconductor package, and we said, okay, we've used this before, let's see what happens on the dream data. So to make our lives a little bit easier in the interpretation, we started just by subsetting to the FGFR pathway and say, let's ignore the AKT for now. There's a clear stimulus of FGFR with the FGF ligand, and there's a drug that knocks it down. So let's see if we can find some relationship between the inhibition and the stimulus to then use to motivate our further solutions. So the idea, I'm going to sort of change plots on you, so just to orient you to the new plot. I'm going from this heat map representation of the plot to now a bunch of line plots, where this matrix is then transformed into each row represents one pattern. So I make just a scatter plot of that pattern. And the dots, the different colors, represent the different conditions over time, where I'd indicated the time by the shading here and the groupings by the color, just to sort of orient yourself. If I go to the BT20 cell line, the first thing that we saw was something that's constant, just sort of a baseline over time. That's something that we typically see in these matrix factorizations. Then we saw something that was transient, and we noticed a huge amount of variability at time 0, which is something that we were sort of struggling with in thinking about the solution. What are we using for our initial condition if we have such large variability at time 0? And this is something we saw in the raw data as well. And the third pattern that we saw that came out of the data that we thought was really interesting is completely unsupervised. What we found is something where in gray represents no stimulation, so just PBS. In green represents stimulation with the FGF ligand, and in red represents serum, so you can imagine that as sort of throwing a stimulus bomb on the system. And then we also have no inhibitor and inhibitor in solid or open dots. And what we see is a pattern that's related to something going up globally due to stimulation of the pathway, where it's down with no stimulus, going a little bit higher with FGF, and then jumping up with serum. But that being said, we don't notice in any of these conditions a real clear separation between treatment and not treatment. If we look at all these, the sort of closed shaded dots represent the treatment, the untreated, and the open triangles, the treated, and they seem to be tracking together. So that didn't really help us either until Sasha Favorov had a great hypothesis and said, well, you've looked at the correlations, you've looked at this. Maybe the inhibition, the reason you're not seeing things in these global scales is that the inhibition is only modifying the phosphoprotein expression of a few direct targets. So again, that's confirmed. And so the first thing that we tried, we didn't submit this to the leaderboard, but we just tried it internally with cross-validation and said, okay, if we have some sort of a time constant model, let's just start with something constant in time. What is that? That's going to be the mean from the training data. If we use that, and we did a cross-validation where we applied it to, we basically trained on AKT and tested on FGFR, we found that our RMS error was almost the same as if we used the true mean of the system. So then we submitted a second solution where what we did was we first interpolated the training data to talk about the missing data issues that were brought up. So we first interpolated each phosphoprotein profile to the output desired time points, and then simply averaged them together across the different inhibitions. And we were very surprised when that solution ended up as top of the leaderboard, and decided to think about it's probably because we're predicting most of the proteins correctly, so we need to focus on the one or two that are affected by the drug next. So we figured we would move on to the in silico data to get insights into how to model that data and the time course data for those proteins, and then come back to the real data. So for the in silico data, we developed what we called a pseudo-linear model. It was basically a linear control ODE model that we added in nonlinearities to enforce that protein expression couldn't go too high or too low. And then we applied the similar ODE model to downstream targets of the proteins. So for that, we need to sort of go through the whole contest of identifying the proteins downstream of the targets for a network, formulating the model, finding the parameters, and then predicting the unobserved values. So what we did is we used a very simple correlation-based approach, nothing as fancy. I would love to try your Granger and more directed model. But what we did is we took formed a correlation matrix, and a sort of a poor man's approach, we find the significantly correlated proteins and then just filter out anything that was indicated as being upstream in canonical signaling pathways from cell signaling. And when we did that, we got these proteins over here. So for we were sort of reassured we got at least proteins that made sense. For EGFR, we got SARC. For BRCA-ABL and SARC, we got CRAF. For mTOR, we got P70, S6K, and Richter. So we were reassured that these were biologically reasonable looking at the network. We then modeled the behaviors with just a simple linear ODE model where we basically say, okay, the protein is going to be going down to something based on who it's connected to in the network. But we didn't have a great way of estimating what was going on in the new proteins when they were treated with drugs in the direct drug targets, because we didn't have a lot of training data for that. So what we did is we just said, let's just estimate for now that they're decaying exponentially over time. So we estimated just a slow 1 hour exponential decay in the target proteins, and then we fit the proteins downstream of that using this ODE model, where we estimated the rate parameters from the data that was there. And when we did that, unfortunately, we ended up scoring worse on the leaderboard than using the average. So that was when we decided to go back again to the in silico data. And we looked a little bit more closely at the data to see what's going on, maybe we're using the targets wrong. So here I've plotted AKT in each of the different conditions over time. So this is phospho-AKT expression in the 2 different phosphorylation sites. And these 2 panels here represent the data under AKT inhibition. And what we've done is overlain, it's sort of hard to see, but if you can see this black line underneath in all the profiles, that's the untreated data with AKT. And so what you see is when we're treating with this supposed AKT inhibitor, we're actually getting an increase in phospho-AKT expression. And we are hypothesizing that might explain some of the discrepancy between the best performances in the in silico and the real data, because if maybe in reality what could be going on is that the cells with lower AKT expression might be targeted preferentially by the drug, they die off, and then what you could be left with is a population that has higher expression of AKT. So you might see that in your data even if individual drugs are being targeted. And that might lead to a real discrepancy between in silico data where you can simulate a knockdown of the protein that you might not be able to get in vivo. So to summarize, the correlation and COGAPS analysis suggested that we were only finding a few proteins that are really altered by the drug, that we shouldn't be doing this globally and predicting these profiles, should just do it for the couple that are direct targets. It's worthwhile that estimating the phosphoprotein trajectories just as the average did pretty well because of that, and that we had unexpected overexpression in response to inhibition in some of these conditions. And I think if any model's going to improve that, going to improve the true trajectory, we need to model that and not just the hypothesized inhibition. The other thing is that we wanted to mention is just uneven sampling might cause this unweighted RMSE, maybe might not be the best metric. It was something we were noticing in the in silico data, that models that we had that were performing better by RMSE when we looked at them visually didn't make as much sense because it was weighing the earlier time points more. So it might be that to get biological understanding better, we might need a different model for this. So we'd love to think about the collaborative round and use a much better network than the crude correlation-based network that we derived. And we'd like to think about what happens if you model both an unexpected increase and decrease in the different species. And does one perform better? You know, it could be by drug, it could be by cell line, we're not sure. So seeing in which conditions does it do better to model an increase, and in which conditions does it do better to model a decrease. And we'd like to include a more advanced ODE model than just a linear model in the future. So thanks.